Next, we'll move on to the third type of cell death, which is necroptosis. So as the name suggests, it is going to be a combination of necrosis and apoptosis. So what part of necrosis is there? So morphologically, it is going to resemble necrosis. That is, it is going to have membrane damage which is associated with inflammation. While the pathways which regulate it are going to resemble that of apoptosis. Not exactly, but it is going to have some pathways which are going to be present. So it is going to be a programmed cell death. So there is going to be some regulation of the cell death. So it is programmed cell death which uses some pathways, and that is the one which is similar to an apoptosis kind of cell death. Okay. So the other name of this necroptosis is programmed necrosis. Okay, or caspase independent programmed cell death. So here there is not going to be any caspase involvement. It is just caspase independent programmed cell death. So this is how it is different from the apoptosis also. And this again can be a result of physiological or a pathological cause. So physiologically, necroptosis can be seen in mammalian growth plate formation. So in the formation of the mammalian growth plate, this necroptosis process is involved. Pathologically also, necroptosis can happen. So in examples like steatohepatitis, acute pancreatitis, ischemia reperfusion injury, neurodegenerative disorders like Alzheimer's, then in CMV infection. So CMV is cytomegalovirus. So cytomegaloviruses are known to produce caspase inhibitors. So whenever there is apoptosis, they can inhibit these caspases and escape from apoptosis. So this against the CMV infections, this necroptosis can act because it is a caspase independent programmed cell death. So what happens in necroptosis is that here again, there is going to be involvement of the death receptors. So death receptors belong to the TNF family, right? So there is going to be a TNF receptor present on the cell, and when there is the ligand, which is the TNF, is going to, so when this molecule TNF is going to come and attach itself with its receptor, it is going to recruit a complex which is composed of three things, which is RIP kinase 1, RIP kinase 3, and a procaspase 8. Okay, doesn't it look similar to the extrinsic pathway of apoptosis? So this is where it is actually resembling the apoptosis pathway. Okay, so there again there was involvement of this extrinsic death receptors, and then there was activation of procaspase 8 in extrinsic pathway. However, in this necroptosis, this procaspase 8 is just forming a complex called as ripoptosome along with the RIP kinase 1 and 3, but it is never going to be activated. Okay, never activated in the necroptosis. That is the difference here. So this complex together, RIP3 kinase, RIP1 kinase and procaspase 8 is together called as the ripoptosome. And this this complex, especially this RIP3 kinase, is going to cause the phosphorylation of this MLKL protein. So this MLKL protein will be there, and that will be phosphorylated by this RIP3 kinase. And this MLKL protein will then cause the membrane damage. It will get relocated to the membrane and cause membrane damage. So when membrane damage is there, it is going to be associated with inflammation. And also this MLKL will cause reduction in the ATP and increase in the reactive oxygen species. So this all will finally lead to the cellular swelling. So cellular swelling is there, membrane damage is there, inflammation is there. So morphologically, how will this cell death look like? It is going to look like necrosis. And its pathway was somewhat similar to it had a regulated pathway, and that pathway was somewhat similar to the extrinsic pathway of apoptosis. So together it makes the necroptosis. Okay.